Greetings, fellow 451 students. My name is Michael Kolarik.
And I'm at Noblett.
Today, we are addressing the topic of biological computing. First, we'll go over exactly what biological computing is, compare and contrast it with traditional computing, cover several of the basic types of biological computers, and how organic compounds fit into the picture. Then, we'll explore the origins and building blocks of these new computers, address a few more of the details of how they are engineered, cover the economical impact, possible applications, and disadvantages of biocomputing.
Biocomputers use systems of biologically derived molecules, such as DNA and proteins, to perform computational calculations involving storing, retrieving, and processing data. Biological computing has moved to the forefront, and the possibility of their development has been made possible with the expansion of nanobiotechnology. Nanobiotechnology can be defined as any type of technology that uses both nanoscale materials, 1 to 100 nanometers in size, and biologically based materials. The implementation of nanobiotechnology gives scientists the ability to engineer biomolecular systems so that they can interact in a fashion that can ultimately result in the computational functionality of a computer. Biocomputers use biologically derived materials to perform computational functions. A biocomputer consists of pathways or series of metabolic pathways involving biological materials that are engineered to behave in a certain manner based upon the conditions that are input into the system. The resulting pathway of reactions that takes place constitutes an output, which is based on engineering design of the biocomputer and can be interpreted as a form of computational analysis. There are three unique types of biocomputers: biochemical computers, biomechanical computers, and bioelectronic computers.
Traditional computers, like the kind that we use on a day-to-day basis, are made of inorganic compounds such as silicon and precious heavy metals including gold, silver, platinum, palladium, copper, nickel, and tin. Biological computers, on the other hand, are made almost exclusively of organic components, namely DNA and RNA, and different types of enzymes. As we know from biology, natural organic components quickly decay. This fact does give an advantage to traditional computers, which, due to their inorganic chemical makeup, last much, much longer. Traditional computers operate in a sequential manner and offer little in the way of parallelism. Pipeline processors, multi-core processors, and high-performance computing, in which many powerful computers operate as one, sometimes known as cluster computing, are the current methods we use to handle many instructions at once. But, due to the nature of biocomputers operating inside of a cell's DNA, the execution of instructions is massively parallel. Each of the countless strands of DNA can be operating simultaneously, each acting as an independent processor or execution unit. Biological computers also greatly surpass our modern computers in energy efficiency. Our most modern, capable supercomputers can complete 109 operations per joule of energy. Contrast this with a DNA computer, capable of completing 2 times 10 to the 19th, or 20 quintillion operations per joule of energy. In addition to this, a biological computer has no clock speed. The speed is only limited by the movement of electrical signals and the RNA interactions within the cell. Currently, the accuracy of these calculations is, however, about 5% less than our current systems.
In most biocomputers, the operations take place within the DNA, which acts as the memory unit. DNA, or deoxyribonucleic acid, is the hereditary material in humans and almost all other organisms. Nearly every cell in the person's body has the same DNA. Most DNA is located in the cell nucleus, but a small amount of DNA can also be found in the mitochondria. The information in the DNA is stored as a code made up of 4 chemical bases: adenine, guanine, cytosine, and thymine. Human DNA consists of about 3 billion bases, and more than 99% of those bases are the same in all people. The order or sequence of these bases determines the information available for building and maintaining an organism, similar to the way in which the letters of the alphabet appear in a certain order to form words and sentences. DNA can be thought of as the source code of life. The slow processing speed of a DNA computer is compensated by its potential to make a high amount of multiple parallel computations. This allows the system to take a similar amount of time for complex calculations as for a simple one. This is achieved by the fact that millions or billions of molecules interact with each other simultaneously. Biochemical computers use an immense variety of feedback loops that are characteristic of biological chemical reactions in order to reach the ability to complete computations. Feedback loops in biological systems take many forms, and many different factors can provide both positive and negative feedback to a particular biochemical process. This can cause either an increase in chemical output, think signal amplification, or a decrease in chemical output, similar to the switch of a transistor in a traditional computer. The factors that cause this depend on how many catalytic enzymes are present, the amount of reactants present, the amount of products present, and the amount of molecules that bind and alter chemical reactivity of any of the enzymes or reactants.
Biomechanical computers are similar to biochemical computers in the idea that they both perform a specific operation that can be interpreted as a functional computation based upon specific initial conditions which serve as input. The main difference comes at the output signal. In biochemical computers, the concentration of certain chemicals serves as the output, but in biomechanical computers, the mechanical shape of a specific molecule or set of molecules under an initial set of conditions serve as the output. The mechanical three-dimensional structure of the product of the biomechanical computer is detected and interpreted as the calculated output.
Bioelectronics encompass a range of topics at the interface of biology and electronics. One aspect of bioelectronics is the application of electronics to problems in biology, medicine, and security. The concept of integrating biomolecules with electronic elements to reach functional devices is attracting research efforts. Bioelectronics is aimed to highlight the world of electronics and combine it with the world of biology and biotechnology. Bioelectronic computers can be constructed to perform electronic computing. Like the other two computers mentioned, computations are performed by interpreting a specific output that is based upon an initial set of conditions that serve as the input. The measured output for bioelectronic computers is the nature of electrical conductivity that is observed in the bioelectronic computer. This output comprises specifically designed biomolecules that conduct electricity in specific manners, which are determined by the initial conditions that are used for the input of the bioelectronic system.
So how did this all start anyways? Researchers at California Institute of Technology laid the foundations for creating biochemical circuits from scratch. They initially conducted experiments using DNA solutions in a test tube. In a conventional computer, logic gates are made with electronic transistors, and these are wired together to form a circuit inside of a silicon chip. These biochemical circuits consist of molecules floating in a test tube of salt water and other molecules. Their next step was putting clusters of these solutions, comprised of DNA and specific enzymes, onto gold-plated chips. Finally, to complete the picture, engineers at MIT have developed biological computational circuits capable of not only responding to data, but remembering input data as well. The unique thing about this is that this biological state machine is living inside of an actual cell. The experiments that were run were programmed into E. coli cells to react to several substances commonly used in biological laboratory experiments. The actual cell behavior programmed by the researchers was the expression of genes coding for the production of different fluorescent proteins representing different colors. With 3 unique inputs, they were able to produce 16 different combinations of colors. This is very similar to the concept of a state machine in traditional computer architecture.
In order to simulate traditional transistors, bioengineers at Stanford University have created the first biological transistor made from genetic materials, DNA and RNA. Named the transcriptor, this biological transistor is the final component that was required to build biological computers inside of living cells. We are now capable of building biological computers that can detect changes in a cell's environment, store a record that change in memory made of DNA, and then trigger some kind of response, such as commanding a cell to stop producing insulin or self-destruct if cancer is detected. Stanford's transcriptor is essentially the biological equivalent of a digital transistor. Where transistors control the flow of electricity, transcriptors control the flow of RNA polymerase as it travels along a strand of DNA. The transcriptors do this by using special combinations of enzymes, integrases, that control the RNA's movement along the strand of DNA. The types of enzymes used are carefully chosen in order to ensure that these biocomputers can be implemented in a variety of organisms. A tiny change in the enzyme's activity, the transcriptor's gate, can cause a very large change in 2 connected genes, the channel, which parallels the concept of signal amplification. By combining multiple transcriptors, the Stanford researchers have created a full suite of Boolean integrace logic, BIL gates, the biological equivalent of AND, NAND, OR, XOR, NOR, and XNOR logic gates. As we know, with only a few different types of gates, we can implement nearly any operation that a computer requires.
Next, I'll go into the process of actually engineering these things. The field of nanobiotechnology provides the means to synthesize the multiple chemical components necessary to create these biocomputers. The chemical makeup of a protein is dictated by the sequence of its amino acids, which are the building blocks of the DNA molecules. The amino acid sequence is dictated by a specific sequence of DNA nucleotides, which are the building blocks of DNA molecules. Proteins are manufactured in biological systems through the translation of nucleotide sequences by biological molecules called ribosomes. These ribosomes group together to transform individual amino acids into polypeptides that form functional proteins based on the nucleotide sequence that is read and interpreted by ribosomes. Basically, this means that someone can engineer the chemical components required to create a biological system capable of performing computations by engineering specific nucleotide sequences to encode for the right protein components, essentially writing the instructions that we normally do in a computer today.
So how does money fit into all this? All biological organisms have the ability to self-replicate and self-assemble into functional components. The economical benefit of biocomputers lies in this potential of all biologically derived systems to self-replicate and self-assemble given the correct conditions. For instance, all of the necessary proteins for a certain biochemical pathway can be modified to serve as a biocomputer and can be synthesized many times over inside of a biological cell from a single DNA molecule. This DNA molecule can then replicate itself many times. This makes biological molecules highly efficient and extremely inexpensive. When you look at the traditional electronic computer that we have access to today, they require manual production, where biocomputers are able to be produced in large quantities from cultures and no additional machinery is required.
These concepts can be applied in a number of areas. In the medical field, we can use this technology to develop cells into differentiated cell types with specific functions thanks to transcription factors. Transcription factors, which we covered earlier, have allowed researchers to program both progenitor cells to become certain types of functional cells, similar to how stem cells are currently being used in conventional medicine. This also paves the way for genetic recombinations, potentially allowing us to cure diseases we never thought possible. Additionally, due to each individual's personal chemistry being slightly different, we should be able to customize medications to a patient's exact physical needs. Another field we can utilize biocomputing in is mass data storage. A single gram of DNA can hold 2 petabytes of data. The obvious implications of this include storing nearly an infinite amount of data in a very little space for much, much cheaper. Complex problems can also be solved much quicker due to the massively parallel nature of these systems. Large, complex mathematical equations and cryptography are 2 of the current areas being experimented on. Finally, one of the more sci-fi-like applications involve new ways of data transmission. Imagine a world where a simple handshake with a friend or colleague allows you to exchange contact information.
Now, while this all sounds good and dandy, but there are a few downsides to biocomputing. Number 1, as we stated earlier, organic compounds have a short half-life and decay over time, which could render the computer inoperable or produce incorrect results very quickly. Unlike the computer systems we are used to, the results are stored in a solution of DNA, and the results cannot be just read on a computer screen as easily. Another entire process must be in place to read the sequence of DNA and check the state of the transcriptors. Number 3, molecular operations are not perfect. Just as errors happen in DNA replications, so can computational errors happen. Most of these strands will have the correct result, currently on an average of 95% of the time, but some will be incorrect and failure rate increases with the complexity of the problem. Number 4, some of the enzymes inputs are uncommon and can be expensive to synthesize in the lab at this point. Since the current implementations require a constant supply of these, can be costly to operate at times. Mike and I thank you for your time, and we hope you learned a little bit about the future of compute.